Hello everyone, I am Faience Auxtero, and today I'm gonna talk about Chloroplast Genome as a class requirement for our crop genetics. When we talk about DNA, what comes first into our mind is that it can only be found in the cell nucleus. However, it is not true because there are also DNA in the chloroplast and the mitochondria. But for today's video, we will be focusing on the genome that can be found in the chloroplast. We know that chloroplasts are specialized organelles that is responsible for photosynthesis, the food-making process. These organelles contains DNA which carry defined subset of the total cell genome. Its size ranges from 120 kilobase pair to 160 kilobase pair that contains approximately 150 genes and 15 of that contains introns. This gene encodes for protein necessary for photosynthetic processes. However, this extranuclear DNA resembles more of the prokaryotes than of the nuclear one, or the one found in the nucleus. As instead of the normal linear helix, this cpDNA is structured circularly, so no histones were associated on this. For one trivia, did you know that mitochondria and chloroplasts have their own genome because they are theorized to be derived from prokaryotic endosymbionts, or commonly known as the theory of endosymbiosis? To continue, chloroplast genome can be divided into three functional categories, including the protein coding genes or the exons, introns, and the intergenic spaces. The latter two, or the introns and intergenic spaces, do not encode proteins as they are the non-coding regions. The chloroplast genome is characterized by 6 repeat families, the 2 inverted repeats, 2 tandem repeats, and 2 dispersed repeats. So for this, from the total of 150 genes, 4 of that are encoded into ribosomal RNA, 24 ribosomal proteins, 34 transfer RNA, 6 genes for ATPases, then genes involved for photosynthesis, and genes for chloroplast RNA polymerase. As most of the chloroplast proteins are encoded in the nucleus, chloroplast ribosomes consist of 52 proteins, but only 19 of that are encoded by the plastic genome or inside the chloroplast. So this process implies that there are still exchange from the nucleus to the chloroplast. Moreover, as being said as one of the important products of cpDNA, it encoded subunits of rubisco, as we know that it is a critical enzyme for catalysis of fixation reaction for carbon dioxide gas in the photosynthesis process. So if you might ask, upon knowing that cpDNA is structured circularly, thus the cpDNA undergoes DNA replication, transcription, and to be translated like normal DNA do? Yes. However, as cpDNA is circular, it does not replicate like the normal process we know, but it utilizes the double displacement loop strategy or the D-loop replication to initiate the DNA replication. The 2 displacement loops begin on the opposite strands of the R replace and begin replicating unidirectionally towards each other until they join to create the bidirectional replication bubble. So, the outside strand of the double DNA is called the heavy strand, and the inner strand is called the light strand because it contains the lighter nucleotide, and the heavy strand contains more of the heavy nucleotide such as the guanine. As it has more guanine, the lighter strand has more cytosine. At this point, the displacement loop fuse forming a theta structure and will produce 4 strands of DNA. And as it interlaps with each other, an enzyme called topoisomerase 3 alpha will separate the old and new double DNA strands from each other and then producing 2 circular DNA strands. So, furthermore, in bacteria, translation initiates and elongates co-transcriptionally, thus ensuring efficient transcription, conferring an RNA stability, enabling timely translation, and maintaining genome integrity by avoiding extended hybridization of RNA and DNA. So this process is called the coupling of transcription and translation. So this process is also proposed for chloroplast gene expression that was based on the early electron micrographs that was interpreted with numerous evidences. However, it is much more complicated because some of the researchers suggested that there are also a post-transcriptional translation for chloroplasts. So this is the overview of the internal and external triggers that cause the regulatory adjustments of translation in the chloroplasts, the mechanism that controls translation, the coupling of RNA and protein metabolism to chloroplast translation, and the localization of the chloroplast translation machinery. So upon knowing that there are also DNA replication, translation, and transcription, how is it inherited? So as plant breeding major, it will be an important aspect to know how does the chloroplast genome will be inherited. So chloroplast inheritance at the turn of the 20th century, there is this German botanist called Karl Correns, and he did a series of genetic experiments using the 4 o'clock plants or the Mirabilis jalapa. The Mirabilis jalapa plants used by Correns came in 3 types. So there is the pure green, pure white, and the variegated or the combination of the 2 color. He was curious because he observed that the green and white branches could appear on the variegated plants, but the variegated branch did not appear on the green and white plants. So, Kearns speculated that some factor in the cytoplasm of the cell, the egg cell to be specific, must be the determinant of the color of the offspring. So this is a model that explains the results of the 4 o'clock crosses in regards to the autonomous chloroplast inheritance, and this is the result of Correns' experiment. So this implies in whatever the trait carried by the male parent that it cannot be expressed, and only from the female parent will be expressed. So this cytoplasmic inheritance is called the maternal inheritance because only the female parent trait will be expressed. So the material inheritance as chloroplast genome is located in the cytoplasm rather than the nucleus. So it is said that during the meiosis, only the egg cell will contribute the bulk of the cytoplasm of the zygote. So as chloroplast is located in the cytoplasm, only the female parent will contribute, or the trait of the female parent will be expressed. And lastly, for the general knowledge of all, as a farewell trivia, these genomes or the chloroplast genomes were first discovered in attempt to explain the non-Mendelian pattern of inheritance in the Neurospora crassa and Chlamydomonas reinhardti. And thank you for listening, and God bless everyone.